The other case will be a frequency generation case, but now not this sort of generating light of a drastically different color, but creating wavelengths that are very near to the laser pulse through spectrally through nonlinear optical effects. So the first technique, this is work that was published in December of 2008, coming from Sunny Shi's lab at Harvard University. And this is using stimulated Raman scattering in order to produce images. And this is done through modulation transfer. So panel A shows the Jablonski diagram of the process. The idea is a pump photon excites an electron to a virtual level, which then would spontaneously decay back to a excited vibrational state, so through spontaneous Raman. But if you send in light that's of that same frequency that matches that transition back to the vibrationally active Raman state, you can stimulate that Raman transition. And if you look in panel B, if you imagine you sent in an equal intensity of light in both the pump and the Stokes beam, if you stimulate this transition from the pump, then what you tend to do is you convert pump photons into Stokes photons, and of course a little bit of heat left in the sample because of the excited vibrational state. So if you look at the output light, you would tend to have a modest gain in the intensity of what's called the Stokes field, and a modest loss of intensity in the pump field. And these changes are quite small. They're on the order of about a part in 10 to the 7. So the only way to be able to detect that kind of change reliably is through these modulation techniques. So the idea is then we leave the pump beam on all the time, and we turn the Stokes beam on and off, as you see in panel C. So it's only when the Stokes beam is on that we would see this modest increase in the pump beam, and we differentiate between those two states with lock-in. So setup is quite simple, shown in panel D there. The Stokes beam is modulated, mixed with the pump beam with a dichroic, sent into a laser scanning microscope, and just the pump beam is measured. The intensity of only the pump beam is measured on a photodiode. And that photodiode signal is amplified in a lock-in amplifier. So with this kind of scheme, intensity and high-frequency modulation, intensity changes on the order of 10 to the minus 6 to 10 minus 7 are readily measured. And on the bottom panel F, that's actually the stimulated Raman scatter spectrum in red of retinol, a important molecule in biology. So here's some microspectroscopy and imaging done by this using the stimulated Raman scattering technique. So imaging, again, lipid uptake in cells, now being able to distinguish between multiple different kinds of lipids, and actually with significantly higher sensitivity than the CARS technique that was discussed earlier. It's also possible to use this modulation transfer idea to directly image nonlinear and excited state absorption. So the molecule no longer has to fluoresce in order to be able to detect it's there. This is work from Warren's lab at Princeton. So in this case, again, there's an optical setup that basically produces a rapidly modulated beam. And then there's a second pulse that's sent in that is not temporally modulated. And you look for that temporal modulation to show up on the non-temporally modulated pulse due to the nonlinear effect. And there's several different effects that they looked at. One would be two-photon absorption, where a photon from each beam contributes to exciting an electron to the excited state. It's also the possibility of excited state absorption, where the pump pulse promotes an electron to an excited state, and then the probe pulse promotes it higher to a higher level state. It's also possible to look at bleaching effects, saturated absorption, stimulated emission, all kinds of these nonlinear optical effects that we've studied for years in solid-state physics could potentially be used for image contrast. One interesting application that Warren's lab has shown is that in hemoglobin, which is a critically important biological molecule, that you can actually distinguish its ligand binding state. You can tell whether it's oxygenated or deoxygenated based on this excited state absorption measurement. So, so on the upper left is this is the strength of signal, the change in the intensity of the probe beam as a function of the time delay between the pump and the probe beam. When the pump is around 800 nanometers and the probe around 650. On the right is the same, except now the probe is at a shorter wavelength. And you can see that when with the long wavelength, I'm sorry, the pump is at a shorter wavelength. With the long wavelength pump, the oxy and deoxyhemoglobin behave about the same. And that's not surprising, because if you look on the bottom figure, you can see that the absorption, the linear absorption of oxy and deoxyhemoglobin has an isobestic point. They cross each other right around 800 nanometers. On the other hand, around 650 nanometers, where we're pumping things on the image on the upper right, you can see that oxygenated hemoglobin has a much lower absorption coefficient than deoxygenated hemoglobin. And so you have a much larger excited state absorption signal when pumping with that wavelength. So by imaging with these two wavelengths, it's actually, by imaging with both of these in turn, it's actually possible to measure the oxygenation of a sample of blood based on a nonlinear optical technique. And the reason that's really interesting, so the images on the top are images of red blood cells taken, the ones in red taken with this method. In the middle is an image stack, sort of like I showed you earlier for the two-photon excited fluorescence, except now a direct measurement of hemoglobin. And you can see this is in the ear of a live mouse, and you can see these blood vessels that are just below the surface. Down at the bottom are a pair of images that were taken with these two different conditions, and you can see different blood vessels show up in one versus the other, and that's because the vessels that show up in the panel on the bottom left have more deoxygenated blood in them, so they're venules. The vessels that show up on the bottom right are a mix of the deoxygenated venules and the oxygenated arterioles. So I'm going to skip the last thing and just wrap up here. So the basic idea is that using nonlinear optical effects gives you a tool to be able to image, to take images with micrometer resolution in 3 dimensions deep into scattering tissues. Many of the classic nonlinear optical imaging methods rely on the generation of a widely separated new color of light, like two-photon excited fluorescence, second and third harmonic generation in cars. And there's a new class of emerging nonlinear optical imaging methods that rely on modulation transfer, or I didn't talk about non-degenerate four-wave mixing, but basically rely on other methods to be able to detect these weak nonlinear optical effects, and provide new tools to be able to study things like the oxygenation state of hemoglobin based on a nonlinear optical imaging method that would allow this sort of 3-dimensional resolution. So one thing that I think is going to be interesting over the next few years is thinking about what other ways we might be able to sensitively detect a nonlinear optical effect and use it as a contrast mechanism for biological imaging. Thank you.